Hi guys, this is Unit 5, Cell Division. This is going to be Segment 1. You're going to learn about cell growth, cell cycle, and something called mitosis. For cell growth, why do cells divide? There are several reasons. The cell becomes too large for the DNA in the nucleus to actually keep control. The cell membrane can't exchange enough materials and waste. So if the cell gets too big, the cell membrane is maxed out because it has to maintain everything inside that cell. So division, it makes more sense, it's more efficient to divide. Cancer cells, it's important to note that they divide and grow at an uncontrolled rate, which is problematic. Why tumors can get so big, because it's uncontrolled cell growth. The cell cycle is kind of a big deal, and I know that this looks like a big confusing diagram, but I think you need to replicate this in your notes because you have to memorize it and understand it. So there's 4 phases of the cell cycle, and if you look over at this diagram as I go through it, it'll make more sense. So you have G1 phase. This is where the cell gets the biggest growth spurt, okay? So the cell is growing, it's functioning, and then it proceeds around the circle into the S phase. And during this time, the DNA inside the nucleus actually replicates itself. It copies itself, and you'll understand why in just a second. And then it proceeds into the G2 phase, and this is just getting all prepped. It's doubling its organelles, making new organelles, getting ready to actually divide. And then this whole piece of the pie right here with all these little slivers, this is your mitosis, the M phase. It's not labeled here, but I would go ahead and label it M. So you have G1, S, G2, M. Those are all the phases of the cell cycle. We're going to specifically get into this piece of the pie for majority of this segment. So, but it's important to also know this arrow is showing the direction that it travels, but it's also showing you that from here all the way around to here is called interphase. So there's like another umbrella name for G1, S, and G2, and that's called the interphase. Then we have the M phase, which is mitosis. Here it is bigger. You can pause the recording so that you can record this in your notes if you need to, but this is the whole cell cycle. This is pretty much everything you're going to need to know for segment 1. So interphase, that one that includes G1, S, and G2, interphase is going to be the longest period of growth. The cell is resting, and the DNA is replicating, and the organelles are doubling in number, and it is the largest part of the cell cycle. Cell division is called mitosis, the M phase. Mitosis is when the cell divides into 2 identical daughter cells, and the key word is identical. These are twins, okay? They match the parent cell perfectly. The same DNA is replicated and formed into the daughter cells. So one cell moves through this phase and becomes 2 cells after the M phase. The 4 phases of mitosis are prophase, metaphase, anaphase, telophase, and I always tell my students just remember PMAT. PMAT, prophase, metaphase, anaphase, telophase. Okay, and then after telophase we have cytokinesis. This is the actual splitting of the cytoplasm, splitting of the cell. So we're going to go through each of those one by one. So it starts with, in PMAT, it starts with prophase. Write all these things down, and I also want you to write down, sketch this cell right here, because this is going to show you what the cell looks like when it's in this phase. And you guys are going to identify real cells, and you're going to try to identify the phase that it's actually in just by looking at it. Yeah, you're going to get that good. So as part of mitosis, the first phase is prophase. This is when spindles form, okay? Spindle fiber forms. Centrioles, those organelles that you learned last unit, they start to move to opposite poles. Right here is your centriole, centrosomes right here. That's what these are. They're stretching and they're forming these spindle fibers. chromosomes become visible. It was chromatin, that loose spaghetti, if you guys remember that vocabulary word. But now we're looking at the X's, the real chromosome structure, when prophase starts. DNA coils around histones forming these chromosome structures. That's prophase. Getting ready. Metaphase, notice how they all line up. All the chromosomes line up single file along the equator of the cell. Meet in the middle in metaphase. Meet in the middle in metaphase. Okay, M's. All in the middle. What are these called? Back there, centrioles. So these are the centrioles, these are the spindle fiber. Notice how the spindle fibers are attaching to the chromosomes also. You're going to see them being pulled here in the next one. So the centromeres divide. They stretch, the chromatids separate and move to opposite poles. These spindle fibers stretch and pull. The centromere is the middle part of the chromosome. See how it's split? So I'm going to go back to that first slide. So you have like your X, your typical X shape. Well, right here is your centromere, and it is going to split. This is where it holds the two sides together. In this next one, it splits. See how it's one leg of the X, and it splits to the other side. Telophase, now we're starting to see some cleavage going, some indent going in. It's about ready to divide. The nuclear membrane starts to form. See this dotted line around it? So you should draw this in your cell when you're making your sketches for each of these phases. See how it's starting to, it's a dotted line, but it'll form a full nuclear envelope, the nuclear membrane, if you guys remember that. Spindle fibers, those are gone now. You have your centrioles there too. Chromosomes unwind, they form chromatin now in that loose spaghetti, and cytokinesis begins. Cytokinesis, cytoplasm cell kinesis division. So this is where your cell divides. Cytoplasm divides, got cytokinesis, and one cell becomes two now. And now you have your two right here. So you have your nuclear envelope fully developed, all the chromatin inside. With animals, the cell pinches inward. With plants, a new cell wall actually starts to form between the two new cells. We know that there is a cell wall in the plants. It's much more rigid, so the cell wall actually forms and divides. I do need you to know about the structure of a chromosome. We did say centromere, and we never really defined it. So here is a chromosome before mitosis. It's a single leg. The chromatid, it's called a chromatid when it's a single leg. Right here is the centromere, usually centrally located, not always. And then once the cell replication happens in the S phase, you have now your full complete X. And so you have 1, 2 chromatids. And then that divides, it splits during the PMAT of mitosis, and you end up with 1 chromatid again. And then cell replication happens. So this is just an overview. There's the new words chromatid, centromere, and chromosome that are introduced to you in this slide. I think that's it.